Hello, everybody, and welcome back. This session is sterility assurance using sterile compounding automation and technology, and it is sponsored by Omnicell. And now I'd like to introduce our next speaker, Kevin Hansen. Kevin Hansen is Assistant Director of Pharmacy at Moses H. Cone Memorial Hospital in Greensboro, North Carolina. Dr. Hansen provides leadership and operational oversight for pharmaceutical compounding and pharmacy perioperative services. He serves as the Residence Program Director for the Health System Pharmacy Administration and Leadership Residency Program. In addition, Dr. Hansen serves as adjunct faculty for the University of North Carolina Eshelman School of Pharmacy. Kevin's complete bio can be found on the Speakers tab in the left-hand menu. But for now, please join me in welcoming Kevin Hansen.
Thank you very much for the kind introduction. I'm going to share my screen here so we can get this presentation kicked off here. All right, so we're going to be talking about sterility assurance using sterile compounding, automation, and technology here today. And very honored to present this information to you all today. So my disclosures are listed here on the slide. Just one note that I am a member of the USP Compounding Expert Committee. However, the views and opinions expressed today are that of my own and not endorsed or affiliated with USP. So we're going to cover three primary objectives during this presentation, talking about some of our influences on our sterile preparations. We're going to be talking about sterile compounding automation and promoting safe and accurate compounding. And we'll finish talking about quality by design approaches for CSPs using such technology. So let's start with the basics, talking about these terms of sterility and aseptic and sterility assurance. I think it's important to note that these terms, sterile and aseptic, are not synonymous, right? In its most strictest definition, sterile is the complete absence of all viable microorganisms. And with this textual definition, we know that this cannot actually be applied to items labeled as sterile because of these irresolvable limitations that we see within the testing that we'll discuss here upcoming. When we contrast this to aseptic, it's more about the prevention of contamination by the exclusion of microorganisms. So many of us that say we perform sterile compounding, really should we change it and say we're performing aseptic compounding or aseptic manipulation. So something to consider. So really what we were going to focus on then is sterility assurance. And this is about adding confidence in our aseptic processing through sound design, reliable equipment, quality materials, effective procedures, and personnel proficiency, rather than solely based off of this independent sampling with sterility testing and some of the microbiological monitoring that we'll discuss. So, with that being said, wouldn't it make sense then to, continue to just test our environments more frequently, every month, every week, every day, every hour? Where do you draw the line? And I think there does need to be a careful balance of this testing frequency, as intensive monitoring may not be warranted. As Ralph described in his presentation, is that even our certifiers coming in to our clean rooms can be vectors for bringing in microbes with all of the different types of equipment and things that can be difficult to clean, if you will. And to note that humans are the ones that are bringing the contaminants into our clean rooms. And so that really needs to be a focus for us as well. And of course, having an expectation of zero contamination at all locations in every aspect of aseptic compounding is technically not even possible nor realistic. So here is a good list of various influences on our sterile preparations. These are all of the systems that are designed to prevent microbes from getting into the sterile preparations. And if they're not working, they can also then promote contamination of our preparations. So we have these broken down by product, equipment, environmental considerations that can include your cleaning, maintenance, personnel practices, your HVAC system, the facility design, your personnel, and even material flow, and the list goes on and on and on. But I think what we're really going to dive into is what is the main driver for contamination is humans. And then, of course, when we look at all of these systems, do you know that every single one of these 100% of the time is working as designed, right? So if we review Murphy's Law, we know that if anything can go wrong, it will. And so are you aware when those things aren't going according to as designed, and having good monitoring practices in place to observe those? And why is this important? If we look over the past couple decades, we see compounding tragedies specifically related to contamination events. Obviously, most notably is the 2012 New England compounding disaster. And, it's important to note that, you see the videos at NECC, they had clean rooms, they had hoods, they had gowned personnel. These, what I call checkbox items, aren't creating our sterility assurance alone. And so we really need to look into how can we provide true sterility assurance in our practices. Also, another observation is that many of these compounding tragedies were related to non-sterile to sterile compounding, as we see within the current context of USP 797 deemed as high-risk compounding. When we think about the different myriads of ways that we can provide sterile injectable products to our patients, there are some that are extremely safe, such as those ready to administer conventionally manufactured premixes that we should be using, as these are the safest products and preparations that we can use for our patients, to using ready to use devices and simple patient specific admixtures to batching, and then all the way down to our riskiest practices, those high risk admixtures. So in the context of a lot of our standards, when we talk about sterile compounding, these are kind of viewed all as equal. And one of the things that I would like us to think about is do we need to treat these completely separate when it comes from a risk perspective and also from sterility assurance? So where do we want to focus in? We can see in this great study here of identifying what are the top sources of microbial contamination. And then as we can see, personnel contaminants is the number one on the list. So what are some of those practices in the aseptic processing environment that can lead to this contamination event? So some of the associated high and very high risk could be the aseptic assembly, aseptic technique used, gowning, material transfer. These are all things that we will address and when we talk about sterile compounding technology and automation. And just to belabor this point is, us as humans, we are covered in this normal flora. We don't think of these as harmful, as they're obviously normal to us. But when these, deemed non-harmful bacteria, fungus, and molds get into areas that they're not intended to, like in our vasculature system, these can certainly cause harm. And even the things that potentially could be brought into the clean room we need to focus on is we know that the cell phone is dirtier than the toilet seat, the toilet handle, and the doorknob combined. And so it's not only the people, but it's also the things that we're bringing in and out of these spaces that we need to be focused on. And if you're following these FDA guidance documents for compounding, we have the insanitary conditions. And there's this inherent focus on this as well, on this concept of that, humans are harbored with bacteria and we're releasing particles. And so they talk about, completely covering up all the skin and the hair, especially when working in those ISO 5 conditions. And those focus areas can be our wrists, our forehead, our legs. Those are areas that we can cover with GARD to ensure that we're not contaminating our environment and thus our sterile preparations. Another thought is not only the garments that we're using to go into the clean room, but even before getting into our anterooms to begin the garbing practice is what about street clothes versus laundered scrubs? So, if your compounding staff are wearing, scrubs or clothes brought in from home, what are they potentially even bringing from home? Do they have cats at home? Are they bringing dog hair? Do they have a smoker at home? None of these things should not only enter your clean room, never enter your anterooms, never come near those spaces. And so having a site laundered program can certainly help to reduce those bio burden. So humans generate hundreds of thousands to hundreds of millions of particles. And this is our skin sloughing off, our hair falling out. And obviously, we normally don't visibly see this, but we need to view ourselves as pigpen in this sense, because when we look at our limits as defined by our ISO classifications, as we know in our direct compounding areas for ISO class 5, is we can't have greater than 100 particles per cubic foot in any given measure. Now, why is this important? As we just said, hundreds of thousands to hundreds of millions of particles, but in ISO 5, we can't have greater than 100. So, if any of your compounding staff have any visible hair, skin, any of that exposed while they're performing manipulations in ISO 5, you are likely not meeting this ISO 5 definition. You may not just know it because you may only be certifying your rooms or hoods every 6 months. So something to be really important is to focus on sterility assurance and not just the testing alone. And, going further into this is looking into the Austin P Encyclopedia of Clean Rooms and this awesome contamination index that really compares not only to the different types of garments that we use into our cleanrooms, but also the levels of activity. You can read certainly some of the FDA 483s and some of our 503Bs. Outsourcing facilities, and they actually will monitor the speed at which the compounding personnel move across the room. And it's for this exact concept: is the more movement, you're going to, shed more particles, and that can get into our environment, into our preparations. And so here we have one of our technicians. You can see in a standard snap smock, which is, unfortunately seen a lot of hospitals across the nation, and comparing that to this membrane coverall. And as you look at these reduction in these particles emitted per minute, is that these two different garments have an exponential reduction as you move from the left to the right to more of these membrane coveralls and cover up more of that skin and hair. So something that certainly should be considered a best practice and that we should be focused on. Other ways that we can contaminate our products is, of course, we have our human vectors, but also water sources. And where do we have a water source? Is with our sinks in our anteroom. And so while we have all of this great garments, and some facilities may be even using sterile garments, well, if you have a sink in the room where that is applying, you are likely contaminating that with water. And where you have water, you have fungus and bacteria as well. And you'll be bringing that into your ISO 7 buffer rooms, which we need to prevent. And so as you saw in the revised proposed USP 797, this option of removing your sink from that traditional ISO 7 anteroom and putting it outside of that anteroom. And in this case, here's an example of entering into an ISO 8. We'll call it a scrub room, where some of the garbing practice can begin. You'll perform your cleansing and garbing, and then enter your dry garbing room to complete the garbing process. And so this is really a quality by design aspect to prevent that microbes from ever entering into our space. All right, so now we want to talk about sterility testing. So in the chapter of USP 1071, they talk about patient safety is best served through the completion of a test that detects microbial contamination prior to product use. So this is interesting because as we read in other statements, we also see that passing a sterility test is not proof that it tested a lot of sterile. So how can we have both of these statements existing at the same time? And so I think it's important, as we'll go through some evidence of what does a sterility test tell us and what does it not tell us, and really how should we use the result of a sterility test moving forward. So here is the USP 71 so for samples for testing. Right now, of course, this is based off of the batch size and the number of containers, and whether it's a small volume parenteral or large volume parenteral. And we can see that we have the listed number of samples that we need to test. This is, of course, destructive testing. So you're going to be testing for sterility in these samples, and the results are then applied to the remaining batch that was held back during quarantine, during that 14 to 18 day quarantine period, if you will. So what does this look like from a statistical perspective? So if we were to look at just the raw statistics of if we had a point failure, contaminated CSP in a batch, what is the probability of this current sampling plan using USP 71 and 797 for these CSPs of 39 or less? What is our probability? And so as you can see, as we increase our batch size, is that our probability of finding that point contamination becomes extremely low. And anything really over 250 units, we can see we drop from 4%, which is already a very low number, down to 2%. And so the conclusion here is that this sterility test is not designed to find that point sterility failure or contaminated product that you have within that batch size. So that becomes really important to look at. There's several other limitations of the sterility test. We know that there's limited organism detection. There's only 6 different organisms that is looked at. Restricted sample sizes, which we talked about. Certain organisms can only grow in certain types of media. The final results of these sterility tests are subjective. It's based off of visualizing turbidity. It is a destructive test, as we talked about. And then we have the minimum of that 14-day incubation period. So if we were to look at this, maybe instead of a point contamination, but maybe a gross contamination rate. So here you can see minimum of 1 in 1,000 all the way to 1 in 2 being contaminated in a batch, is that our sensitivity of finding having that failing sterility test with those sample sizes, our ability increases. And so, should we be viewing the sterility test as really only identifying, did some type of gross contamination event occurred, as you have a leak in your HEPA filter, where it was, one of the drugs that underwent sterility through filtration, did that filter fail? I think there's many things that could indicate, but the statistics really show that probably is the best case. When we look into previously known as sterility assurance level, with our probability of a non-sterile unit, we see these different values published in the literature. Pharmaceutical sterilization, using terminal sterilization of being, no more than one non-sterile unit in every 1 million produced. These numbers are completely arbitrary and really don't have a particular scientific rationale. It's actually legacy coming from the food canning industry. So I think, just noting that, but there's certainly some literature out there estimating, even for aseptic processing, of course, these numbers can't be proven, of potentially even contamination rates of 1 in 1,000. And so again, when you apply some of these realistic values of probability of contamination, our sterility test is not designed to find those point failures. So, one of the thoughts that has come up is, well, what if we just test more samples? So, maybe we, the sterility test requires us to test 10 samples. Why don't we test 20? Why don't we test 50? Why don't we test 100? Will this help to increase it? And so there's a great article by Moldenhauer and colleagues published of looking at this from a Poisson probability distribution perspective. And the Poisson probability distribution is really looking at that these contamination events may be independent and not related to one another. And so looks at the distribution of that throughout the batch. And so we have our probability here. We have our constant E. And then we also have lambda, which is our mean contamination rate. And so then if we take 1 minus the probability to the nth power, the n being the samples tested, that can give us our probability of failing that sterility test. So, and in this case, really the size of the batch is irrelevant. So if we were to apply this, we assume a frequency of contaminated units in a batch is 1 in 1,000. So lambda here is 0.001. So if we test 10 samples, the probability is around 1%. If we double our samples to 20, you can see that our probability of failing only goes up to 2%. Even if we were to test 50 samples, it only goes up to just less than 5%. So again, just testing more samples may not be the right answer. And also note that these are destructive tests. And now we're going to have extensive drug waste through this. So this may not be the right piece. And lastly is the end results are very subjective. And we're looking and comparing the sample to a positive control and negative control and trying to figure out, is this turbid or not? My eyes may be very different than yours. This could be also problematic. Even if you were to pick that non-sterile, failing result, if your eyes can't tell it, you're not going to reach the right conclusions. So in this document, the pharmaceutical CGMPs for the 21st century, they actually encourage the use of modern microbiological testing methods that are more accurate and more precise. And I would even go further and say that, end results that are objective and not subjective and based. USP 797 allows for use of these tests using a validated alternative method that is non-inferior. So you do need to perform that testing before using these other methods to the USP 71 tested as outlined in USP 1223. So there are numerous different rapid microbiological methods out there. Some are growth-based, some are non-growth-based. One example is ATP bioluminescence, which is actually amplifying ATP in a sample and actually using a luminometer to actually measure the amount of light detected from that ATP using an enzyme reaction. And so again, that can be an objective test instead of a subjective test. So now, as we kind of take a step back and look at our different practice settings for sterile compounding, we have our 503As following our USP 797 standards. We have our 503Bs that are following these modified CGMP practices outlined in these guidance documents. And then our manufacturers are following the full CGMP. I think it's important, while not required nor practical in this full sense, really as 503A compounders, we really should not ignore what we can learn and adapt from our CGMP principles. This may be a transition for many of us as very type A pharmacists. We love our checkbox mindset. Tell me what I need to do and let's check that box. But we need to shift our mindset and thinking more of an engineering mindset in our design. We need to design it. We need to prove our design works, and we need to demonstrate that it continues to function as designed with no defects. So we'll go into comparing many of these. So we've already stated that personnel are the most significant contributors to microbiological risks. So what are our goals to combat this? So as outlined, we have our goals of what if we separated our personnel from those aseptic environments, right? If we know that we're the ones contaminating, let's get us out of there, right? How can we also limit the interaction with all of these sterilized components in products to do this? So one way we can do that is using compounding automation. So here we have pictured as an IV robotic device. It performs all the aseptic manipulations within a closed restricted access barrier systems with some limited access ports that we'll talk about, but it allows us to meet those goals. It reduces or eliminates personnel interventions and thus the personnel-borne contamination. It's separative technology to eliminate, to the extent technically possible, those human source contaminants. And going back to the CGMP in the 21st century, and mind you that this was published back in 2004, right? This guidance encourages the adoption of new technological advances by the pharmaceutical industry. In particular, the guidance underscores that advances in automation and isolation concepts offer in protecting the exposed sterile drug product during its aseptic manufacture, right? So we have our FDA, our manufacturing colleagues, really encouraging the use of these types of things. And so we have some opportunities for our other practice settings. Then, of course, what is the evidence of really showing that these do have reduced microbiological burden? In USP 1116, a microbiological control and monitoring of aseptic processing environments, they compare these ISO 5 critical zones really from a manual perspective and find that we should expect recovery rates of 1% or less with our attainable current methods for testing. Contrasting and comparing that to advanced technologies, which they define as isolators and closed restricted access barrier systems, are used at not more than 0.1%. Yes, a 10-fold reduction, as we see from these ISO 5 critical zones of microbiological samples should have recoverable microorganisms, right? Of course, as we described, these are never going to be zero, but we do see that reduction because of those goals that are achieved through this. And when these devices are running, the direct intervention by gown personnel is disallowed during that process. And so over the next couple of slides, we're actually going to break it down and show how those can function to prevent that from occurring. So here is the face of an IV robotic device in a restricted access barrier systems. This is a primary engineering control. It is equipped with a HEPA filter with ISO 5 laminar airflow in a vertical fashion coming from the ceiling down. This is located within a clean room suite in the ISO class 7 buffer room. There's a syringe port for adding and removing syringe. There's a syringe cap port, a drug valve port, a bag port, and the door to this device is completely locked during operations, again, to prevent contamination of that critical zone. As we look inside the device, we can see several different aspects here. So we have, modules for syringe dosing. We have modules for labeling. We have a module for, rotating the bags and selection of bags. We have a robotic arm that is programmed and performing all the aseptic movements, and not pictured here, but then on the front end of the screen is the syringe capper module. And the robotic arm can be programmed such that it is not disrupting that laminar airflow or blocking any of those critical transfer sites. So really important. And lastly, we'll talk later about our UVC disinfection lamps that are contained within. And so just like our primary engineering controls, surface sampling is performed on key areas throughout the direct compounding area within the RABs, and these are certified. We perform air samples, just like any primary engineering control. And it's important to know that this robotic arm is programmed to be positioned in a way that each movement does not block the airflow. A robot is only as good, of course, as how we program it. However, it does not fatigue like humans. So maintaining this laminar air in these systems can be done to a higher degree of confidence continuously, and we can prove that through the various types of testing that are there. So when we look at the technology today, our current standards that are out there for compounding, we know have an inherent focus on manual practices. Now, technology and automation is viewed as a best practice, as we see from our leading safety organizations in the world, and they reference to them. But there's a focus on accuracy and precision and ingredient verification. While those things are really important, again, we kind of are missing that aspect of how do we promote sterility within these cases. And in some cases, some of our technology may actually go against sterility as we see, increased use of touch screens with our, compounders that are wearing sterile gloves. So certainly a problem. So in our current state, we know that we have this proposed revision of 797, which certainly does not prohibit use of this type of technology. I certainly think there's opportunity to discuss, more clearly, where do these things fit into our practice? And we know that there's the ISMP sterile compounding guidelines, which encourage us to have a strategic plan for implementation of this type of compounding. And so we can see that we certainly have some opportunities. And a few years ago, there was held an international IV robotics summit up at the Cleveland Clinic. And, one of my observations as we got to hear from compounders in different countries throughout the world and their use of these IV robotic technologies is that in the United States, we're a little bit behind comparison to our European colleagues when it comes to use of IV automation in many different settings. And the overall emerging theme that we captured from this summit is, there's benefits of these IV robots compared to manual compounding. And we see that with precision, with accuracy and the documentation, the safety features, and then of course, those sterility advantages that we're really focused on. So now we're going to shift into our last concept on this quality by design for sterile processing. So what is this? It is a systematic approach to development that begins with predefined objectives and emphasizes product and process understanding and process control based on sound science and quality risk management. And we'll talk about how this applies to the use of compounding automation and technology. And our goal is let's optimize contamination control with a particular focus on the microbial risk impact of our personnel. All right. We've seen the evidence showing that our personnel are the ones that are contaminating these products. What can we do to minimize that from a design perspective? Looking into the CGMP for the 21st century really kind of gives us a roadmap for what this looks like upcoming, as we know that quality, safety, and efficacy are designed or built into the product. Right now, I think this is a really important statement within this document, is that quality starts from the front end, not from the back end, right? So this finished product testing is still important, right? But we can't say that just the testing alone that we have, quality processes. We really have to build in quality from the front end, have process verification, and we will discuss what that can look like. These IV robotic devices are obviously very complex. There's a lot of moving parts. There's a lot of programming. There's engineering involved. These devices certainly are not plug and play. I don't want to kid anybody that these devices can just be wheeled in and start making CSPs. These are critical devices. Proper installation and testing of these devices is of the utmost importance, especially anytime we're involving patients on the other end. So work with your IV robot vendor to ensure that these devices are installed and tested really using a CGMP style manner that includes the DQ or the design qualifications, showing a piece of technology has a compliant design from the get-go. Can it even be successful as designed? Are they performing the IQ installation qualification? This includes, is it set up? Is it connected? Is it working as planned and as defined in your design qualifications? Next is the OQ, the operational qualifications. Is it working as intended in all aspects? And doing lots of performance testing on this can be really important. And then lastly, leading into this is that performance qualification, the PQ, is showing it delivers overall desired performance based on acceptance criteria. So, as part of your program is defining, how do we define accuracy? How do we define precision? What values do we assign to this? And is the device able to meet that on a consistent manner and having data to prove that? Performing media fill tests and showing that it can maintain sterility. There's a lot of things that need to be done in the front end before these devices should be used. And again, these are aspects that we can learn from CGMP and learning from our manufacturers for safe use of these devices. Also important is that after going through that extensive installation process is that the work doesn't stop there. Even when they start producing, is you have to have continuous quality improvement plans. And as we start to think more like engineers, is how can we incorporate failure mode and effects analysis, or FMEA? And so, this is really looking into, each of our processes and saying, what's the failure mode or what could go wrong in this process? Our failure causes, why would this failure happen? And then the failure effects, what are the consequences of this failure if it were to occur? And for each of these steps in the process, as you go through this spreadsheet, is you're trying to determine from a numerical value from 1 to 5 or 1 to 6, depending on what tool you're using, to start is, what is the probability of this event occurring? Is it something that's going to be extremely likely or extremely unlikely? The severity of an event. Is the severity associated with the event? Is it, not going to have really any impact? Is it more, is it a documentation thing, or is it going to, be something that's going to be extremely critical and cause, extreme harm or even death to a patient? And the last piece, which I think is really important, is what is our ability of the system to detect that an event occurred, right? And when we think about our current sterile compounding practices you may have in your 503A setting, this is why we have restricted beyond use states. It's very difficult to determine if we've contaminated a sterile preparation. We certainly can't see it. We're obviously not performing sterility tests on every preparation. And so this is why, risk is increased for our 503A practices, and we have those restricted BUDs because our ability of our system to detect it is extremely low, if not impossible, right? So once you have the numerical values for each of these, you'll go ahead and multiply those out, and it'll really then give you a priority of where do we need to focus on from a corrective action and preventative action standpoint. And then making sure that those are tagged with somebody responsible, having the due date, and how do we measure it to make sure that it actually was fixed, if you will. And, this is a great chart. I use this very frequently. It comes from the National Patient Safety Foundation. As we look at instilling actions from any types of findings in our CAPA planning or FMEA, how many of us have heard maybe in our organizations of, we just need to do more training, or we need to send out a memo or an email to everyone. We need to add another double check. We need to add, alerts. It seems that, we that's kind of our go-to gut instinct about how we solve some of these. But when you look at this chart, those are some of the weakest actions that we can actually take, right? So how do we shift from the left end of the screen to the right end and start instilling maybe more intermediate or stronger actions? And so with regards to compounding automation, we can see that many of those things actually address it from a strong perspective. So having engineering controls, forcing functions, simplifying processes, standardizing on equipment and process, having tangible involvement by leadership, devices with usability testing, and even some of those architectural plan and changes. So these are all things that would be viewed as strong actions when you're going through to fix some of these issues. Another thing from a quality by design perspective is one of the things that we found in our organization, before any types of events even happen, right, is how can we prevent them from happening in the first place? And one opportunity for contamination was actually in the pre-assembly before the compounding even begins. We have a syringe and we have a needle. And, at start, this was a risk, as there could certainly be a contamination point during this assembly, especially if you're transferring it from one hood to another hood, that can become very problematic. So one option to significantly reduce, if not eliminate that opportunity, is purchasing pre-assembled syringes and needles where available. Another area that was identified is within the sterile syringe caps. The sterile syringe cap packaging is very awkward and it requires very specific technique to get these out of the packaging and get these into the IV robotic device without contaminating them. And so, simplifying the process, have point training, demonstrating how to do this in a correct manner, can become essential because, again, all of your other systems could be working. The IV robot may be working perfectly, your clean room and all the monitoring, but if you can't get these sterile caps into the device in a sterile and compliant way, you may have a contamination failure. So this is why it's important that we look at every element in this process that we have for sterile compounding. Another option, we talked about humans bringing things into the clean room, but what also about our drugs and our materials that we use? So, there's been some evidence reported out by ISMP of actually having hospitals having aerosolized fungal contamination of our clean rooms, and it was actually found to come from the wholesaler totes, right? This is a very, this could be a very problematic thing. And so really looking into your cleaning and disinfecting process as you're bringing materials and drugs into your clean room space. How are you doing that? What chemicals are you using, right? So in these totes, they actually found cladosporium, aspergillus, penicillium species, right? So if you're using a germicidal wiping process or a sterile alcohol process, you're not even going to be effectively killing these spores and these fungus. Incorporating a sporicidal into the process before these get into your buffer room can really be important to make sure that this doesn't occur, right? We need to take the adverse experiences of others and not say that this doesn't apply to me, but taking it seriously and making sure that we can prevent these things from getting into our space. And next, we talked about the UVC disinfection process. So this is a UVC radiation. It's germicidal. It effectively eliminates viruses, parasites, bacteria, right? It almost sounds too good to be true, right? Well, looking in to make sure that you have the right wavelength can be really important from a germicidal perspective. Making sure that you are using it at the right length of time, that you're providing enough radiation to actually get those claims. So there's certainly some testing in there to do to make sure that you're having the right dose. But I think the important piece here is that these should only be used to augment manual cleaning and disinfection protocols and not used as a sole replacement. As we know, especially inside these robotic devices, there's a lot of surface area, but the UVC is only going to be effective on areas of where those rays of light are going to. And so if there's areas where are not getting hit by the lights, in some of these areas, then those are going to be missed opportunities. So we need to ensure that we're coupling this with manual cleaning and disinfecting practices as well. Another quality by design piece, as we talk about the different types of compounding. So within our organization, we do patient-specific adult compounding, patient-specific pediatric and neonatal compounding, and then we have this non-patient-specific batching operation as well, is, talking about the busyness of each of these clean rooms as we're making stat medications for our emergency room and getting these patient-specific doses up for, meeting code sepsis is we really found an opportunity of we need to separate these as much as we can. Even with adult and pediatric doses, we don't want to have these mix-up errors. And so we actually provided completely independent clean room suites within our organization for each of these operations. So really important. All right, so now we're going to end on a case study about how, where do we use IV robotic automation to produce ready-to-administer syringes. So we're going to start by this sample situation. So, and you're a 503A hospital setting, let's say. So your organization purchases ready-to-administer rocuronium syringes from a 503B. This is used to improve patient care, safety, decrease bedside preparation. There's a lot of evidence of using these ready-to-administer preparations. You receive an email from your 503B vendor that due to increased demand, all products will now be shipping 3 to 4 weeks late. I can't see folks, but I'm sure there's some head nodding going on. This is unfortunately all too common. So due to this continued supply chain disruption and impact to patient care, your organization evaluates, should we use IV robotics to repackage rocuronium into ready-to-administer syringes? So it's also important to note that simply just by having a robot device doesn't mean that you can just compound anything you want under the sun. You need to do things in the right way, right? Especially if you're using extended stability and beyond use states. So to start, you have to have validated methods. So going through having system suitability, looking at calibration curves, accuracy, precision, specificity, you've got to start with validating your methods first. Then doing a robust stability study and not just potency as others have presented during this conference of the importance of looking at degradation, force degradation, and being able to isolate those different analytes, if you will. And really the methods during your stability study too is making sure that it's being performed on these IV robots, doing triplicate lot testing, and looking at some additional tests that can include appearance, pH, particulate matter, endotoxin, sterility, container closure, and having a robust extended beyond use state stability study prior to preparing these can be really important. If you were to apply the upcoming revised USP 797, here we have category 3, is we start that this is performed in a clean room suite. The compounding method, this is all aseptic manipulations, no terminal sterilization is occurring. is performing or passing a USP 71 sterility test or equivalent. All the starting ingredients and packages are all sterile, and then meeting all of those category 3 requirements, we can see that we can have up to a maximum of 60 days room temp or 90 days refrigerated, of course, limited by what was found in the stability study as well. From a workflow perspective, these are compounded and prepared on the IV robotic automation device. It's really considered sterile repackaging. Preparation of a visual inspection of these products as they're coming off and observing the labels and making sure they scan within the system. Taking those subsamples and shipping those off for testing and taking the rest and locking those up into a quarantine process. And then you can see here is using the ATP bioluminescence rapid sterility testing of having that objective end result from a sterility testing. And in about 6 days or so, we'll be able to view those final results, perform one last visual inspection prior to release, and then we release those for patient use. And so you can see having a really defined and safe process prior to these going out. Now, why consider doing this? And so if you look at the current, setting with 503Bs, is that, we have over 6,000 hospitals, in the nation here, but we have only a total of 74 503B facilities. We know that the demand for these products are not being met by our 503Bs, so there currently exists many gaps of providing these products to patients. Obviously, they're performing CGMP, which is superior to the USP 797 standards and works for manufacturers, but we can see that many of them are not following this. As FDA goes through and inspects these sites, it's very common that a 483 will be issued and citing of areas where they're not compliant with CGMP, and we see this over 93% of the time. And then, of course, many of our 503Bs, 82% of them, that is, actually intend to compound sterile drugs from bulk substances. And when you go out and observe these 503Bs, they are all for profit. You can see that they are highly manual processes with minimal to no automation. Now, there's certainly some that have invested into automation, and that's great. But what incentives are there for the others that have highly manual processes to use compounding automation and technology? So I want to end on this slide and really, take a step back from all of this sterile compounding in our hospitals and all of our pharmacies, and actually just imagine with me that you're walking into the grocery store, you're picking up your gallon of milk, and you're leaving there, and you notice that you have this self-checkout device. And as you're going through it and you really start thinking about, maybe you got work on the mind, you're like, wow, there's software guiding me through this process. There's soft stops, there's hard stops, there's barcode scanners, there's not one, but 2 gravimetric scales. And in some cases, they're recording the whole video of your whole transaction. In some cases, they maybe even have an image or object recognition by placing it on the scale and recognizing that it's an apple and not a banana. If you're buying an electronic device, it can have a security tag, a deactivation, and even to pay, you could even tap your phone and have a wireless payment. So I'm going to end by asking this rhetorical question is, are you okay with the fact that there is more technology and automation using your grocery store to get your gallon of milk than there is currently in your pharmaceutical cleaner? Thank you so much for your time in presenting this out today.